It's a great pleasure to welcome you all here for the first Wednesday seminar of the year, and a really great delight for me to introduce Olga Kondrashova to you all. Olga was a postdoc in my lab. She began her PhD with Paul Waring across at Melbourne Uni in 2012, and I was on her PhD committee. So it was a really great for me to attract her to my lab as a postdoc in 2016. And she has been in my lab for 3 years, and she's going to present a tour de force for you of the work that she's accomplished in the time that she's been in my lab. As you know, we make ovarian patient-derived xenografts in my lab, and that's what she's predominantly been working on in one of our strengths, that of PARP inhibitor therapy. So it's a story that really mixes in patient-related outcomes with some very basic science work, which she'll describe. She's completed her time in my lab, very sadly for us, and has moved on to the laboratory of Dr. Nick Waddell at the QIMR in Brisbane. So not only has she got much better weather than we have, but she's gone into the exciting world of bioinformatics. So she will continue to be supervised by Dr. Tony Pappenfuss and working on some material coming out of our lab, in addition to a whole new world. So you're going to hear about all of that, and I welcome to the podium Olga. Thank you.
Thank you, Claire, for a kind introduction, and I'm really delighted to give a talk here today. So I will be talking about improving patient selection for PARP inhibition in ovarian cancer. And as Claire mentioned, I'll be talking about the work that I've done in Claire's lab here at WeHi in the last 3 years, and I've had some wonderful times here. And you can see Claire and me looking at microscope slides without a microscope. So I'd like to give you a bit of introduction to ovarian cancer. It's a devastating disease, and it's the most lethal gynecological malignancy worldwide, with over 60% of women who are diagnosed with ovarian cancer being diagnosed at advanced stage. And survival rates have remained relatively unchanged over the last 30 years, despite all the new knowledge that we're gaining in the area, with the 5-year survival remaining below 50% for all stages. And these poor survival rates are largely due to, firstly, lack of disease screening and early detection, but also generalized approaches to treatment, and finally, also due to inadequate preclinical models. So it's important to understand that ovarian cancer is not a single disease. In fact, it's characterized as multiple diseases combined under one name. So just looking at cell of origin, there's multiple subtypes there, with epithelial subtype being the most common, so found in 90% of cases. And then just looking at histological subtypes of epithelial ovarian cancer, we can see over 5 different subtypes, all with different drivers and different biology underlying these cancers. So high-grade serous ovarian cancer is a subtype that I'll be talking about today, and it's the most prevalent one, where 70% of cases are identified as high-grade serous under epithelial group. And then to take one step further, the high-grade serous ovarian cancer can be also classified by molecular subtypes. So there's 4 subtypes just looking at expression base. And the key features of high-grade serous ovarian cancer are, firstly, ubiquitous p53 mutations, so they're found in 100% of all cases, and which leads to high genomic instability. And you can see below is a circus plot of one of the high-grade serous cancer cases that we have. I'm showing the copy number track and lots of translocations detected throughout the whole genome. And another important feature of high-grade serous ovarian cancer is that up to 50% of cases can have defects in HR pathway, homologous recombination pathway. And these include mutations in BRCA1 or 2, but also methylation of BRCA1, RAD51C, and then mutations in other HR-related genes. So what is this homologous recombination? Well, it's a pathway, it's a double-strand DNA break repair pathway. It's highly accurate, and it only is active during S phase. And it's a complex pathway. I'm not going to go into the detail about how pathway works, but there's a number of key players. And importantly, loss of a critical HR gene, so by mutation of BRCA1 or BRCA2, for example, can lead to HR deficiency and consequent sensitivity to PARP inhibitors. I'll call it PARPi therapy. So it's so-called mechanism of synthetic lethality, where a cell proficient in HR pathway, upon PARP inhibition, will survive, but cells deficient in HR pathway will undergo cell death. And so there are a number of theories of how PARP inhibitors work, and the most prevalent one today is that PARPi can cause PARP trapping on DNA and block progression of replication forks. And there have been a number of trials demonstrating efficacy of PARP inhibitors in BRCA1 or 2 mutant ovarian carcinomas, including this study 19, which is a pivotal trial for PARP inhibitor olaparib. And here you can see that patients with germline mutations had significantly longer progression-free survival for olaparib compared with the placebo. So you can see here. But also, it's important to note that some of the patients in the germline wild-type group also derived some benefit from olaparib. So, suggesting that in this wild-type group, there are some PARPi-sensitive tumors, which may harbor other HR deficiencies. For example, somatic BRCA1 or 2 mutations, or mutations in other HR genes, or methylation of BRCA1 or RAD51C. And it's important to further stratify these patients for PARPi response to identify which patients will benefit from PARPi. Now, other research questions that we can ask is, how can we prolong and improve responses to PARPi, even in the responding patients? And also, can we explain the mechanisms of PARPi resistance? And so to study this, we need adequate models. And as I mentioned before, there's a lack of these in high-grade serous ovarian cancer research. So 3 of 5 most used cell lines, highlighted at the bottom in the red box, do not actually resemble high-grade serous ovarian cancer at molecular level. So we need to choose cell line models with caution. And also, as Claire already mentioned, I was quite privileged to work in her lab because there have been over 20 patient-derived xenograft models established in Claire's lab of high-grade serous ovarian cancer, all of which retain major genetic characteristics in regional tumor. Also, PDX drug responses correlate with patient responses, and it's a good renewable resource for preclinical analysis. So with this introduction in mind, I'm going to move on to the major part of my talk. And here I've divided it into 3 parts: mechanisms of PARPi resistance, PARPi response beyond BRCA1 or 2 mutations, and then also combination treatments to enhance PARPi effect. And I will present a few stories on each aspect, and some completed, some ongoing. And hopefully I will demonstrate how we're working towards improving patient selection for PARPi. So I'm going to begin with part 1 of my talk, mechanisms of PARPi resistance. And I'd like to introduce this concept of pre-existing versus acquired resistance. Where in pre-existing resistance, there might be a subclone that's already has the resistance mechanism, and then under the PARPi, or not PARPi, just any treatment selection, you will enhance the clone, and so the progression will develop eventually. Where acquired resistance is de novo resistance that emerges under treatment pressure and also leads to progression. But understanding whether the resistance is pre-existing or acquired is very important to help us decide on the best treatment strategies. And there have been a number of mechanisms of PARPi resistance described in literature to date. Some have been validated just in vitro, some have been seen in the clinic. And today I will focus on two of these. So the work that I'm going to present involves Ariel 2 Part 1 trial. It's a trial to show the efficacy of PARP inhibitor recoparib in platinum-sensitive high-grade serous ovarian cancer. Here, tumor tissue from patients was sequenced using next-generation sequencing, and they were classified according to either BRCA1 or 2 mutations, and then if they were wild type, they were divided into two groups, LOH high group, and in this case, I'm talking about genome-wide loss of heterozygosity, which is a marker of HR deficiency. So this LOH high group would be likely to have HR deficiency. And then the third group, LOH low group. So unlikely to have HR deficiency. I'm showing the results of the trial here. You can see that the blue BRCA mutant group performed better with a longer progression-free survival compared to the LOH low group in green. But also you can see on the right that duration of response in responders of LOH high group was in similar length to the BRCA mutant group. And so pre-treatment and post-progression biopsy pairs were collected for 12 patients, mandatory pre-treatment biopsies and optional post-progression. And Foundation 1 sequencing was performed on these 12 tumors, with 6 of 12 tumors having mutations in HR pathways, so either BRCA1, RAD51C, or RAD51D. And we focused on these 6 cases to study the acquired resistance to PARPi. And I will present 3 of these in detail. So the first case involves a patient with a primary germline RAD51D frameshift mutation, which caused premature termination of the protein. And this patient had 2 prior regimens of chemotherapy before entering the trial. And throughout the duration of the trial, there were 3 lesions that were being monitored. 2 were responding, and 1 was progressing throughout the duration of the trial. And at the pre-treatment point, a liver lesion was biopsied, and the primary mutation was detected in that. At a progression point, the progressing splenic lesion was resected, and at the same time, a biopsy was taken from a liver lesion. And in the responding liver lesion, we also saw this primary mutation. However, in the splenic resection, we saw a secondary mutation that sat on top of the primary mutation and restored the reading frame of the protein, so restored the correct termination of the protein. So we called it secondary mutation. And these secondary mutations have been reported in BRCA1 and 2 as a cause of resistance to platinum chemotherapy and PARPi by a number of people. In fact, it's the most commonly described mechanism of resistance. But it hasn't been reported outside of these 2 genes. So we wanted to study this mutation in detail to see its role on PARPi resistance. And as the sample was collected on the other side of the world, we didn't have access to viable materials, so we had to take it to in silico models and in vitro models to study it. So firstly, we performed predictive modeling, and unfortunately, the primary mutation, because it caused nonsense-mediated decay, the model was too unstable to actually do. But I'm going to show you a simulation of 6 wild-type RAD51D molecules wrapped around double-stranded DNA. And as we zoom in, you will see the red residue is the region that is substituted with lysine in the secondary mutation. And you can see that the RAD51D proteins are interacted with double-stranded DNA when placed in virtual proximity. And when we modeled the secondary mutation, we saw that that interaction was retained. suggesting that the revertant protein may retain some, if not all, function. We also wanted to model this in vitro, and we went to use high-grade serous ovarian cancer cell line. And here we show that a frameshift mutation in RAD51D causes loss of the protein, while the secondary mutation, you can detect the protein. And also, I'm showing here a RAD51 foci assay, a surrogate marker for HR deficiency. So if you're detecting RAD51 foci, it means the HR pathway is active, while if you're not detecting them, it's inactive. So secondary mutation caused the formation of foci. I'm not sure how well you can see this, while the frameshift mutation resulted in lack of foci formation. We also tested rucaparib and cisplatin response in vitro in these models, and we saw that the secondary mutation in orange resulted in resistance to both drugs. Now, the second case that I'm going to present involves the patient with primary germline RAD51C mutation, and here it's a nonsense mutation. And this patient had only one prior regimen of chemotherapy. And 3 lesions were monitored, and all 3 had response to the therapy. However, after 11 months of being on the trial, the patient developed a enlarged groin lymph node, and that was biopsied, and we detected the primary mutation in there. But also we detected 4 independent secondary mutations, all of which sat on top of this primary mutation, and all of which restored the full length of the protein. So once again, we wanted to study these mutations in detail to see their role in causing resistance. First of all, what we noted was a high degree of heterogeneity. So we had 2 biopsies collected from the same lymph node, and we sequenced both of them. And you can see that in the first core, all 4 mutations were present, all 4 secondary mutations were present, while in the second core, just going from one section to another, there's a big shift in allele frequency between the mutations that are present, so that's showing a high degree of heterogeneity. And we were lucky enough here that we had access to viable materials, so we transplanted it into mice to establish a PDX model, but unfortunately, we selected the piece of tumor that only had one of these secondary mutations, so we couldn't study all of them. So we once again went to use cell line models to model these mutations, and we saw a similar thing to what we saw with RAD51D, where these secondary mutations resulted in resistance to rucaparib, while the primary mutation resulted in sensitivity, and also performed 2 assays for HR activity. And they showed the same thing, that the primary mutation resulted in HR deficiency, while the secondary mutations restored HR pathway, and so caused resistance to PARPi. Now, the third case that I'm going to present to you is BRCA1 mutated case. And the primary mutation, again, was a frameshift. First secondary mutation that we detected was a classic mutation where you get a deletion, which restores the reading frame of the protein. But the secondary mutation was quite interesting because it was a splice site downstream of the primary mutation, and it resulted in an alternative isoform of BRCA1, shorter, partially functioning delta 11q isoform. And this isoform has been previously reported to cause resistance to platinum and PARPi. But this was reported in vitro model only, and here we see it in the actual patient. Now, going back to all 6 biopsies that were tested with HR mutations, we actually detected secondary mutations in 5 of these 6 tumors. And all of them did the same thing, where they restored the reading frame of the protein. So to summarize this part of my talk, We first of all confirmed that RAD51C and D mutated cancers can respond to PARPi therapy. We also showed that this resistance through secondary mutations is not limited to BRCA1 or 2 genes. And we highlighted the importance of trialing early delivery of PARPi or combination strategies. And you might remember that in RAD51D case, the patient had 2 prior regimens of chemotherapy, and one of the lesions was never responding, and that's where we detected this secondary mutation. So it's likely that this secondary mutation was developed during the prior regimens of chemotherapy. And in fact, a recently published study, only last year, SOLO-1 trial, showed that maintenance of PARP inhibitors in newly diagnosed ovarian cancer patients shows a much better response. So more than 50% of patients can receive long-term remission. But we still need to study how common are these secondary mutations in PARPi-treated samples, especially in the early treatment setting. And future trials might provide the answer, but we need post-progression biopsies collected, or at least tumor cell-free DNA collected to answer this. So moving on to the second part of my talk, PARPi response beyond BRCA1 or 2 mutations. As I mentioned before, HR defects are quite common in high-grade serous ovarian cancer, with up to 50% of cases having these defects. And I've already talked in detail about BRCA1 to RAD51C and D mutations. But I would like to shift your focus to BRCA1 methylation. It's the second most common defect in high-grade serous ovarian cancer after mutations in BRCA1 and 2. It's detected in about 10% of all cases. But it has been quite an enigma in the world of HR deficiency and PARPi response. First reports of BRCA1 methylation around 20 years ago in breast and ovarian cancer. And there have been numerous reports showing that methylation leads to reduced mRNA and protein expression of BRCA1. And BRCA1 methylated cases have the same gene expression signature as BRCA mutated cases. So on the right, I'm showing the TCGA plot of, in the blue, you can see the methylated cases with the reduced expression of BRCA1. So all of this data suggests that BRCA1 methylation should really cause HR deficiency, and hence should lead to PARPi sensitivity. But it's interesting that BRCA1 methylation is not associated with improved survival, like the BRCA1 or 2 mutations are in ovarian cancer. And again, this is TCGA data. And also there are mixed reports on BRCA1 methylation and PARPi response. So in Ariel 2 Part 1 trial, there was a weak association with PARPi response, but in another trial where they were looking at long-term responders in study 19, there was no association with response duration in these patients. So we wanted to get some clarity on the role of BRCA1 methylation being a useful whether it's a useful biomarker in predicting PARPi response. And we were very lucky because we had quite a number of preclinical models available to study this. We had 4 PDX models, all with BRCA1 methylation, and 2 were chemo-naive, 2 were post-treatment. And then we also had 2 cell lines, one derived from the PDX model and one off-grade cell line just available widely. So we were able to assess PARPi response in 3 of these 4 PDX models. And also we had the PARPi response data for the patient of the 3rd model. And the response was variable. So there were 2 models that were completely refractory to both recoparib, which is a PARPi, and cisplatin. There was 1 model that was responsive, and also in one of the patients we saw a durable partial response. And when our collaborator, Alex Dobrovic, when he looked at the methylation closely, so here I'm showing the methylation-specific high-resolution melting curves, we saw different states of methylation. So in the top two models, you can see the green peak overlaying the methylated control. But in the bottom two panels, you can see that the green peak has actually has two peaks, one overlaying the methylated control, one overlaying the non-methylated control. And because this assay was human-specific, and we knew that PDX models were 100% tumor pure from the human point of view, we could confidently say that this different state of methylation wasn't coming from contaminating human stroma. Alex also looked at this methylation using another assay, methylation-specific droplet digital PCR, and we saw exactly the same story, where two of the models, the two models that actually responded to PARPi, had 100% methylation, and the other two had 50%. And so we explained it by the 100% being homozygous methylation, while the 50% being heterozygous, where one copy was methylated and the other copy was not methylated. So our hypothesis was that homozygous BRCA1 methylation would cause complete silencing and hence would lead to HR deficiency and would sensitize cells to PARPi, while this heterozygous methylation would result in resistance. So we went on to testing this. First of all, we looked at RNA expression of BRCA1 in these 4 models. In fact, this is the data from the patient samples, so there's a bit of contaminating human stroma here. But we've also done the same on the actual PDX samples. And you can see that I've highlighted in the blue box is homozygous models, and in the orange is the heterozygous. The homozygous samples had reduced BRCA1 expression, while heterozygous did not. And we saw similar results by Western blotting. So the homozygous did not have expression of BRCA1, while heterozygous did. And it makes sense from the point of view of just general biology, because that's what we expect to see with BRCA1 and 2 inactivating mutations. Then we also looked at the RAD51 foci formation assay, which is the HR surrogate marker. And here we saw, again, that the heterozygous model had RAD51 foci formation, and so it had active HR pathway, while the 2 homozygous models did not have foci forming. In the 2 cell line models that we looked at, luckily we got one of each, so we got one heterozygous cell line and one homozygous one. We saw the same thing. So looking at PARPi response here, the off-grade cell line that had heterozygous methylation did not respond to PARPi recoparib, while homozygous one did. Now we wanted to validate these findings in patients. So we went back to Ariel 2 Part 1 trial samples, and again, we were very lucky to have access to these samples. And they went to Alex's lab, and he tested them for the methylation using his quantitative assays. However, it was a lot more challenging to analyze here because of contaminating normal stroma that was coming from a human. And so we chose to focus on high-confidence homozygous group that we could clearly establish and see how good of a biomarker it is for predicting PARPi response. So in this first plot, you can see it's a waterfall plot showing the degree of tumor volume change in the trial. You can see that all the green bars, which represent homozygous samples, all of them had significant response, while the yellow bars are the heterozygous cases, and the blue ones are the ones that lost methylation completely. And this is summarized in this plot here, where we saw significant differences for homozygous BRCA1 methylation compared to the wild-type group. And we also looked at the survival, progression-free survival. And here, while not statistically significant, we saw that the median progression-free survival for homozygous high-confidence BRCA1 methylation group was very similar to the BRCA1 or 2 mutated group, while all other methylated cases sat next to the wild-type group. So to summarize this part of the talk, for the first time, we explain how BRCA1 methylation can be used to predict PARPi response. And this is quite important because around 10% of patients will have this methylation. We also showed that if we want to use BRCA1 methylation as a biomarker of response, we need to use quantitative methylation analysis. Otherwise, we just won't get that knowledge. And then I didn't present this data here today, but we showed that BRCA1 methylation loss can happen after exposure to chemotherapy, and loss of methylation of just one BRCA1 copy is enough to cause resistance. And so we need to validate this finding in more patients, and especially in the early setting before resistance can develop. And we can attempt to do this through this EMBRACE clinical trial that we involved in doing the translational work for the trial, and it's currently open. And this study looks at response of patients to olaparib, which is another PARP inhibitor. And here, patients with HR deficiency that are not germline BRCA1 or 2 mutations, because we know quite a lot about those patients already. Patients with either breast or high-grade serous ovarian cancer are tested in this study. And we have funding to collect pre-treatment and post-progression biopsies in high-grade serous ovarian cancer so we can study the methylation in detail here. Now, I would like to move to the last part of my talk, and here I will present a few short examples of some of the work that is very much ongoing or just developing now, sort of future directions point of view on combination treatments to enhance PARPi effect. So why would we combine treatments? Well, even in the best context of the first-line treatment, we know that not all patients are going to be long-term responders. And cancers do adapt and develop resistance. And some of the HR-deficient cancers have inherent mechanisms of resistance. And there are a number of combination trials that are currently being tested in the field. So one of them is enhancing HR deficient phenotype by either downregulating HR pathway or through introducing additional DNA damage. Then also impairing cells' ability to stall the cell cycle, so to repair those PARP-trapped stalled replication forks. And another exciting combination strategy is immunotherapy, and finally, other unrelated targeted therapies. So the first example that I'm going to give you is on enhancing HR-deficient phenotype. And this is the work done in collaboration with Elaine Saanich and Rick Pearson at Peter Mac. And they're looking into this drug called CX-5461, which is an inhibitor of RNA polymerase. And Elaine and others have shown that this drug can induce replication stress and activate DNA damage response. So in theory, it should enhance PARPi response. We've tested this drug in 2 of our HRD models, and here on the left, I'm showing BRCA2 mutant model, where we see some response to single-agent olaparib, PARP inhibitor. But we also see a combinatorial effect of olaparib and CX-5061 combination. And on the right, I'm showing a resistant olaparib lineage of BRCA1 methylated PDX. And here, we can see single-agent activity of CX-5061. And this is important, potentially important for the cases where there is inherent mechanisms of PARPi resistance in the patients. Another combination is immunotherapy with PARP inhibitors. It's a SOLACE 2 trial that Claire and her collaborator Magdalena Plibansky established. She's an immunologist at RMIT. And it's a complex study design. I won't go into detail about the way the study was designed, but I'll highlight a couple of key differences between this study and a number of other combination trials that are being performed currently of PARP inhibitors and immunotherapy. So firstly, patients are selected very early on in their disease at the first asymptomatic progression point, so rise of CA1 to 5 marker, which is a biomarker of ovarian cancer. And this is important for the priming stage of the trial, where low-dose cyclophosphamide is used, together with olaparib, is used to prime, to enhance response to immunotherapy. And by priming with this low-dose cyclophosphamide with olaparib, we hope to increase the number of responders that will respond in the next stage to combination therapy. And so I will hopefully remain involved in the translational aspects of the study, so performing the genomics analysis on this study. And here we're planning to do whole genome sequencing or whole exome sequencing and RNA sequencing where possible on archival tumors, which are the primary samples, and then also optional pre-treatment and post-progression biopsies. We will also be collecting tumor cell-free DNA up to 26 time points because of the fortnightly PBMC collection for 12 weeks. And Magdalena's lab will be looking after the PBMCs and the immunology parts of the study. And so the questions we're hoping to address with this study are, first of all, how to select patients that respond to combination therapy. So identifying genomic signatures that might predict the response. Also identifying functional, potentially functional neoantigens. We also want to look at mechanisms that could cause resistance to the combination. And finally, we want to assess if tumor cell-free DNA can be used to monitor resistance and response in patients. So that's SOLACE 2, and now for the last bit, I'd like to make a jump to the start of my talk, where I mentioned that Claire's lab has over 20 PDX models of high-grade serous ovarian cancer. And what we're currently doing is we're planning to look at whole genome sequencing data. So we've already generated data. We want to identify molecular targets in other pathways, not related to HR pathway, by analyzing this data, and then trialing these potential combination therapies in vivo PDX models. We have 15 models that have been sequenced, some chemo-naive, some post-treatment, and then there's some HR-deficient and some HR-competent, with a range of PARPi responses from refractory to sensitive in both groups. And this work will be done, or is done currently, with Claire's supervision and then Tony's, and Nick is currently supervising me at QRMA Berhofer. But there are also other questions that we want to answer with this whole genome sequencing analysis. Firstly, we want to perform in-depth whole genome characterization of these PDX models to better understand them from every point of view. We also want to look at how much genomic tumor diversity is lost by passaging PDX into these cell lines. The cell lines were established from these PDX models. And thirdly, can we identify mechanisms of unexplained PARPi resistance in these specific models? So with this, I'd like to wrap up my talk by just summarizing the key points on each part of the talk that I did. So for the mechanisms of PARPi resistance, we identified secondary mutations in RAD51C and D. We also identified BRCA1 delta 11 Q isoform in a patient as a mechanism of resistance. And we found that heterozygous BRCA1 methylation leads to resistance. In terms of PARPi response beyond BRCA1 or 2 mutations, we saw the mutations in RAD51C and D cause sensitivity to PARPi, and also homozygous BRCA1 methylation. And finally, for combination treatments, we're looking at a number of trials here. So CX-5461, SOLACE-2, which is an immunotherapy trial, and finally, we look to identify other targets through whole genome sequencing analysis. So with this, I'd like to acknowledge everyone involved in these projects, because they're translational studies. There's so many people involved from all over the world. But really, I'd like to say special thank you to Claire, my supervisor, and also Matthew, who's given a lot of mentorship through the years. And then people in bold who were major contributors to some of the papers that we produced. So Holly, Christy, Monique, Tony has been a great supervisor for the whole genome sequencing analysis, and hopefully he'll continue to play a role in that. And Ming is a person from Clovis Oncology who has been involved in the RAD51C and D secondary mutation story. Elizabeth Swisher, our collaborator at the University of Washington. Alex, who's done all the BRCA1 methylation analysis, and everyone else involved in the trials. And of course, last but not least, Nick, who's taken me into her lab to do some of this future work with her. And I would like to also thank women with ovarian cancer and their families and friends for participating in studies like this. Thank you.
I could start by just asking how you think you're going to move to this next stage of expanding from the minute detail that you've been working on to looking at whole genome sequencing, and how you might have designed some of those studies.
Yeah, it's quite a shift from looking at one gene to going to whole genome sequencing, but we will be looking at, I'm currently looking at, the data was just not mature enough to present here today, but I'm looking at mutational signatures that underlie each of the models, and just the bulk tumor burden and mutations in other pathways and copy number changes and structural variations, all sorts of things.
Other questions?
Great talk. I must have missed it. But did you mention what the timeline is of the heterozygous BRCA1 mutation when you treat it with PARP inhibitors? What's the timeline of developing that mutation?
Oh, the resistance. We don't actually know that yet, because the samples that were collected, we could really only test for the patient samples. We could only test them at the point, a single point in time. So they either already had resistance, this heterozygous methylation, or they didn't. So we can't say for certain how long it takes.
Direct correlation showing that once the BRCA1 mutation occurs, then it can never respond to the PARP1 inhibitor.
I guess that's the idea, that once they develop this resistance through either secondary mutations or through loss of methylation, they become HR-competent tumors again, and they won't respond to PARP inhibitors. And also, they're less likely to respond to other chemotherapies. So platinum chemotherapy.
But the question is, so the mutation was not induced by the PARP inhibitors?
Don't know that yet.
You know for some of them that they didn't
Yeah.
act, and they did have an option.
Yeah, so for some of the, for the PDX models, we know that they didn't develop it during PARPi therapy. We know we tested some of the archival material at chemo-naive stage and then post-chemotherapy, and we know that they lost methylation somewhere at that point. But it would definitely be an important question to answer, at which point do patients lose methylation or gain these secondary mutations?
Thanks for a great seminar. You mentioned at the beginning the problem with ovarian cancer being a lack of early diagnosis. Has there been any progress in this area? For example, is circulating tumor DNA or cells, is that proving useful in early diagnosis in this disease?
I'm not really the best person to comment on this, perhaps, because I'm not involved in that part of work. But yeah, from memory, I think, I don't know, maybe Claire would be best to comment on this.
So there's certainly a lot of work being done looking at TP53 mutations. I mean, at the moment, it would be hard to know where such a signal was arising from. There is being work done, particularly in Germany and the US, looking at p53 mutations in pap smear type fluid. For p53, it would be most likely to be arising from the fallopian tube, not from the cervix, for example, and so work is ongoing there. It hasn't reached the stage of large tertiary type confirmatory studies. So right at the moment, there isn't anything at a mature stage.
Could you elaborate on the low-dose cyclophosphamide and how you expect that to induce immunity?
Yeah, so I don't know, sorry, I'm not immunologist, so probably not the best person to comment on this either. But
So we would just, Magda Plabansky has been working on this for quite a long time, and so she's got a lot of detailed work showing the way in which T regulatory cells are changed by low-dose cyclophosphamide itself, and certain other chemotherapeutic drugs can also alter which populations of T cells are present in tumours in response to that. So the actual amount of cyclophosphamide and the cyclical changes that you need are still to be really proven, and so that's what the 12 weeks is all about. So we'll be taking very frequent PBMCs and looking at changes just as we can look at in the marker in the blood, and then the outcome, will really be what happens with the subsequent response to the combination therapy.
So it is the metronomic dosing?
It is, yeah.
So with a lot of the PD-L1, PD-1 studies, there's been no definitive biomarkers for predicting outcomes. So I'm just wondering how the bioinformatic analysis in this study is going to try and tease that apart further. So peripheral blood mononuclear cells, when you're looking at PD-1 expression on CD8s, is showing some correlation. I'm just wondering how you're looking to progress further beyond previous trials.
We'll be looking at a whole range of markers, and that's why we've got fresh cells being couriered real time on the day from all over Australia and New Zealand. So $2 million was to pay for the trial, and $1 million was to collect the samples and courier them and analyse them by fax fresh. So thanks to AstraZeneca and $400,000 from Oasis, which is a philanthropic branch of ANSCOG's fundraising, we've got the money to actually acquire all of those samples, and now we're fundraising to actually do the analysis for the rest of the translation. So you hit the money, hit the nail on the head. It's the money for sample collection that was really expensive. So there'll be a huge amount of biomarker analysis done.
I was wondering, with the idea of combining olaparib with an RNA polymerase I inhibitor, do you imagine that to be more like the idea of combining two relatively independent mechanisms like immunotherapy and olaparib, or closer to the synthetic lethality that comes from BRCA1 and olaparib, which is trapping the same
Yeah, it's sort of a bit closer to the second, I think. So inducing the DNA damage response. There's a number of theories of how CX-5461 works. One of them was the one that I outlined with Elaine. Another one is forming DNA quadruplexes that result in DNA damage. So yeah, I think it's just enhancing that phenotype. So probably a bit closer to combining chemotherapy with PARPi, I would say, because some of the chemotherapy can be DNA damaging as well.
Any more questions? Great, just thank me in joining Olga for a really fantastic